Hi everybody, can you hear me?
Yes, sir.
Yes, sir.
Okay, I'm just how many of you are there? I'm not in that MS Team screen. I'm in the PPT screen. How many of you all are there?
So currently 8.
Only 8. Okay, we'll wait for a
Now
couple of
9.
minutes.
Now
Okay,
9.
I have to go back there. Some people are waiting in the lobby. Okay, we will start in the next 2-3 minutes. We'll wait. We'll see if more people join, and otherwise we'll start at around 10:05. We'll start. Okay, guys. Hi, everybody. I think there are enough of us. I think we'll get started. So before we start, I'll just introduce myself, and then we'll have a quick round of introduction of all of you, so that I know how many of you have done biology or have not done biology up to 12th grade. So we'll do a quick round of introduction. You can just tell your name and the course that you're associated with. Give me a second. Some people are waiting in the lobby. Okay. Okay, fine. Now, okay, my name is Vivek Perumal. I'll be your course instructor. I'll do all the lectures for this course. My contact information is listed on this slide. Uh, you can contact me by email or by phone call if you have questions. Anytime, actually, no worries. You can just call me, or you can contact me by email if you have any questions at any point of time. Uh, I will try to do 5 to 6 on Tuesdays as office hours. If any of you specifically need a long session, we can do it that time. Um, that's me. So what we'll do is we'll just go around. Give me a second. I'm just going to call names as it appears on my screen. If you introduce yourself by just saying something like about yourself and the course in about 30 seconds or less, then we'll get started with the lectures. Ada?
Good morning, sir. Good morning, everyone. My name is Ada Svetlana. I'm a part-time PhD student, and my background is in microbiology, and I'm currently working on diagnostic development.
Okay. Agrima?
Hi everyone, I am Agrima and I'm a final year BTech student from the Department of Biochemical Engineering and Biotechnology. And I'm currently working on developing temperature sensitive plasmid for Gaudonia.
Ashish?
I'm Ashish Prasad and I'm from Department of Chemical Engineering and in my final year.
Good. Faranullah?
Good morning, everyone. My background is biotechnology. I did my bachelor's in medical lab sciences from Jamia, and I will be working with Kusuma School of Biological Sciences. in deciphering the role of microtubules in chronic leukemia.
Gayatri?
Good morning, everyone. So I'm in the second semester of PhD with Zaire. It's a collaborative project where I'll be working on therapeutics and virology. My background is biotechnology. Thank you.
Hayati?
Good morning, sir. I am Khyati. I'm a BTech third year student at Biotech department.
Biotech, okay. Malvika?
Good morning, everyone. I'm Malvika Ramani. I'm currently in my second semester of MSR at KSPS at IITD, and I'm in Dr. Anita Roy's lab.
Milan?
Good morning, sir. Good morning, everyone. I am Milan Kumar Lakshman. I am a full-time PhD student at KSBS. I have joined in this semester under Dr. Manideepa Banerjee, and I have done my MSc in agricultural biotechnology.
I see. Prakriti?
Good morning, everybody. I'm Prakriti. I'm a PhD student in the joint UKIDAR PhD program. And my host department is the Department of Biochemical Engineering and Biotechnology.
Pranay? Sure.
Good morning, sir. I am Dr. Pranay. I am from medical background and joined PhD in School of Interdisciplinary Research.
Sure. Thank you. So, Radhika?
Good morning, everyone. I am Radhika. I am from the third year and I am from the biotechnology department.
Biotechnology department, okay. I see. Ratnesh?
So, good morning, sir. I am also from biotechnology and biochemical engineering and biotechnology department. I am also in third year and BTech.
Sure. Sanas?
Good morning, everyone. I'm Sanaj Agarwal. I am a final year undergraduate in biochemical engineering and biotechnology department. I'm doing a project in nanopore sequencing, and I'm interested in healthcare and diseases. Thank you.
Sure. Shreya?
Good morning, everyone. My name is Shreya Gupta, and I've just joined KSBS as a full-time PhD student under the supervision of Dr. Ashok Patel, and my master's was in biomedical science.
Siddharth?
Good morning, everyone. So this is Siddharth. I'm a student in KSPS only. So I'm currently working on brain regeneration in zebrafish, and my background is BTech biotechnology.
Sure. Vishantan?
Good morning, sir. I'm Vishantan, and I'm a current third year undergrad student in the Department of Biotechnology and Biochemical Engineering, and I have a profound interest in genomics.
Great. Okay. That makes it complete. So I think most of you have some background in biology, which is also good. So maybe I'll just get going with my slides. Let's see. Okay. Here we go. So today will be just an introductory lecture. I'll try and introduce you to viruses and the basics of virology, and also give you what is the course about and the policy for evaluation and audit, etc. I'll give you all that in today's class. Okay. Okay, so what are viruses? Try and make this class proactive by just answering like whenever you feel like, stopping me whenever you feel like. That makes this class much more interesting and proactive and keeps everybody's attention in the class. So any of you want to try and define what are viruses in your own words? Guys, go right ahead. I'm sure all of you know what viruses are.
So I would say viruses are a collection of like DNA or RNA and proteins, and which can infect different cells and perform functions which they couldn't perform earlier.
Okay, that's not a bad definition. So like whatever you said, we start from the DNA or RNA, right? Everything, pretty much all organisms that we know of, they have genetic material, and like one of you rightly said, for viruses that could be DNA or it could be RNA for some viruses as well. In other words, viruses can have DNA or RNA as genetic material. This is also an exception because typically most of the organisms that we know of contain only DNA as the genetic material. So that's the genetic material. So what's outside the genetic material? What protects the genetic material of viruses? Those of you who are familiar with it.
Yes, sir. There is a protein coat which protects the genetic material.
That's correct. It has a simple protein coat, and this protein is encoded by the virus genome, DNA or RNA. It's encoded by the virus genome, and that protects the genome. So, do some viruses have an additional layer up outside the protein coat? And if so, what is that called?
It is called capsid.
No, the capsid is the protein coat itself. I'm asking what's outside the protein coat.
So I guess the viral envelope.
That's correct.
Envelope.
So that's the virus envelope. Not all viruses are enveloped. Okay, that's important to remember. It's fairly simple. By the end of the course, you'll know that very well, and you'll also understand why. So not all viruses are enveloped. Only some viruses are enveloped. But if a virus is enveloped without the envelope, it's useless. So if a virus is enveloped, it needs the envelope. And what are these envelopes made up of? These envelopes are made up of proteins that are encoded by viruses, and also they contain small parts of the host cell membrane from which the virus pinches off. The envelope is partially derived from the host cell membrane. When the virus infecting a cell pops out of the cell, after replicating inside the cell, it can pinch off some phosphoproteins and some proteins from the host cell membrane along with its own proteins, along with its own envelope protein that it encodes, and that forms the envelope. Now, what sizes do viruses come in? Guys?
Well, viruses are like very small particles. It can be, I guess, 10 nanometer to 100 nanometer.
That's good guess. And there is one thing that we didn't discuss in this whole thing about viruses. This is exactly pretty much whatever you told, right? DNA or RNA is the genetic material. There is a protein code that protects and may be needed for virus entry. Some viruses are enveloped, and that contains virus proteins, and it also may contain host proteins and host phospholipids, etc. An envelope may be important for virus entry and pathogenesis. We'll look at it later. And viruses come in this size range usually. I'll tell you a little bit more about the size so that you can remember it better. I'll give you an example. And the last line is something that we didn't discuss. It's an important one. Viruses are obligate intracellular parasites, which means that they don't really have a life outside the host cell. They can only replicate inside host cells, so that's why they're called obligate or compulsory intracellular parasites. Now, what are viruses? We can have 1,000 definitions. I just picked one from somewhere, I don't know where, but it says viruses are a piece of bad news wrapped up in a protein. That's pretty much what viruses are. So if you look at how are viruses different from bacteria, most of you are much more familiar with bacteria, E. coli, and many other bacteria. You're much more familiar with, you can rattle off a lot of names, and all of us have been infected with bacteria as well. How are viruses different from bacteria in your opinion?
Sir, I have a question.
Go ahead. Go right
Sir,
ahead.
you said that like viruses are bad news. Sir, is it really that if viruses doesn't exist, we don't have any use of viruses, the ecology, the environment, and everything will function well?
No, I mean, see, this is a definition. You can't take it at the literal sense. See, bad or good can be put in perspective. See, if you get infected with a mild variant of COVID, it can be good news because it protects you from any further infection with more dangerous variants of COVID for at least for some period of time. So this is not a textbook definition. This definition is supposed to be something that you can easily remember and relate to a virus. It's not something that you'll put on paper. You understand what I mean?
Yes, sir. Yes.
Okay. Okay, now how are viruses different from bacteria in your opinion? What are some differences? Can each one of you say 1.3 or 4 points at least?
So the main difference we can say that virus is a free living. It can live outside the host cell also, but the viruses need a obligate host. It cannot replicate outside the host cell.
Okay, give me 1 second. Seems like somebody is trying to join. Let me see if I have to let somebody in. No. Yeah. Okay. Give me just 1 second. Okay, so how are viruses different from bacteria? They're not necessarily free living. It's questionable if they have a life outside. If they have life outside the host cell, that itself is questionable. So they're not clearly free living because they may just be retain their infectivity outside the host cell for a few hours. That we still don't know. We are still debating whether viruses are alive or they don't have a life. We are still debating that. So we can't call them free living. What are some other obvious differences between viruses and bacteria?
I think one is the size range.
Okay, very good. One is size range. Very good. What else?
So absence of metabolic networks.
Yeah, go ahead.
So absence of metabolic networks and pathways.
Sure. Basically, I would rephrase that as bacteria are able to do whatever it takes to replicate themselves independent of a host cell because they don't really have organelles per se, but they have ribosomes and they have a lot of metabolic pathways wherein they can actually produce the amino acids and they can also produce the nucleotides that are necessary for bacterial replication. But viruses don't. They completely rely on the host cell's nucleotides and amino acids to make their own proteins and to replicate. So that is one. What else?
Viruses are acellular. There is no point of like cell in the virus, but in bacteria we do have cells, bacterial cell and cell wall and all.
Sure. So I've kind of summed up the same thing. Viruses could be made up of RNA as well. Bacteria is typically made up of DNA. Viruses require a host cell to replicate. They're much smaller in size. It's even it's debatable whether they are living or not living. So that said, what are the sizes of viruses? What sizes do they come in? Now, if you look at the whole light blue colored cell that we have, that's the E cell, and that is approximately about 7 microns in diameter. Now, if you look at bacteria that we know of, right here you have E. coli, and this side you have a streptococcus, which is a circular bacteria. They're roughly 2 nanometers long and 1 micrometer and 2 micrometers long. And if you look at the smallest bacteria, they're called Rickettsia, and they're about 300 nanometers or zero point 0.3 microns in size. Now, if you look at the largest virus, that's the pox virus that's here. That's almost the same size as the smallest bacteria, almost comparable size. But they are difficult to see using a normal microscope, which is why you'll need an electron microscope to look at these organisms. Now, then I have a variety of viruses. This is herpes virus, and then we have the bullet-shaped virus, which is rabies virus here, and then you have HIV, and we have a variety of viruses. So, for example, this number 8 here is poliovirus, which is a very important virus, and you can see it's only 30 nanometers in size. So this is the size range that viruses come in. They go all the way from 20-30 nanometers to about 300-350 nanometers. and the biggest viruses are comparable in size to the smallest bacteria that we know. Okay, so this is just about virus sizes. Now, viruses, there are some unconventional agents. Just because we are talking about viruses, I thought I'll tell you one more thing about viruses or something related. There are organisms called viroids, and these are made up of RNA only, and they don't code for a protein, which means basically they are just a small piece of non-coding RNA that is single-stranded and circular usually, and they don't code for a protein. And so far, the viroids that we know of, they all infect plants, and we don't know any other viroids that are capable of infecting human beings. So this is just a lateral distractor from the main thing. Now, many of you, in fact, all of us have had virus infections. Now, what do virus infections lead to in terms of disease symptoms? What is the outcome of virus infection? Anybody wants to take a guess? What is the best thing that can happen to you if you had a virus infection? Maybe we'll start there. Guys, are you guys with me?
So artificial
Yes, sir.
infection in case of transfection, that we use certain lentivirus and adenovirus to transmit our gene of interest into the
No,
particular
I'm
cells.
saying actually natural infection, nothing to do with cell culture. I'm talking about when you or me get infected with a real virus in real life. What is the outcome? What all things can happen, and what is the best thing that can happen to you?
Development of immunity against the virus.
Okay, there are many ways of looking at it. What you said is all correct. So, excuse me. So, many times virus infections could be asymptomatic, which means you may not even know that you have virus infection. That can happen a good number of times. Having said that, viruses do cause symptoms. More often than not, they do cause symptoms. And in children, I mean, this is US data, they've documented that at least 7 physician visits or doctor visits are caused by virus infection in children every year or annually. And it accounts for almost majority of absenteeism is because of virus infections. Now, if you think of, can anybody tell me a virus infection that you've had? Then we look at category by category and see what all can virus infections lead to.
So seasonal flu.
Seasonal flu. Okay. Let's say you had seasonal flu. What will happen? You had some mild symptoms. What happens after 7-8 days?
So we feel better and yeah, that's it.
You fully recover, right?
Yeah.
So that's called, so good example. So this is called acute self-limiting infection. What do I mean by that? All this is written, I think, in the slide. But before I show what is written there, we just want to discuss it, and then you'll connect to it much better, I think. So seasonal flu, I get infected, I have some symptoms, mild or severe. After 6-7 days, usually most people recover. So that's called acute self-limiting, which means acute means everything is done in a small period. That's acute. Self-limiting means your body's immune system is able to take care of it and resolve the infection. That's called acute self-limiting infection. Can anybody give another example of a virus infection which does not manifest something like what we say, what we looked at in common flu?
Sir, measles.
Okay, measles. What happens in measles? There also, measles is also typically acute self-limiting, although it can have long-term sequelae. We'll come back to it. Any other better examples where the body is not able to clear the infection?
HIV.
Sir, HIV and COVID-19.
Not COVID-19. COVID-19 is typically an acute self-limiting infection. It can be
So
fatal.
rabies and HIV.
Say that again.
So rabies.
Rabies is also fatal and it can have sequelae. HIV, somebody said HIV. How is HIV different from common flu in terms of the consequences?
So it actually attacks the immune system itself. So like
That is
again, it's
okay.
harder for the body to counteract.
That is perfect, but is it acute self-limiting? Is it short term and can we clear the infection?
No, sir.
So that is
No,
called a
sir.
persistent disease because we get infected and we remain infected for life, and that type of virus infection is called persistent disease. No, like some of you said, some virus infections can be fatal. For example, rabies. If you don't get good immunoprophylaxis, the rabies infections are almost always fatal if you don't get the vaccine and the immunoglobulin. We'll talk about it later, but if you don't get that, rabies is fatal. So viruses can cause fatal disease. How many of you have had chicken pox? Anybody?
Yeah, had it once when I was young.
So even I've had it. So for the two of us, we fought the virus, we became better, but the virus is still in hiding in some parts of our nervous system. It never leaves the body. The immune system is not able to fight it off from some parts of the nervous system. The virus remains in hiding there. It's not replicating. So that is called latent infection. Now, what does it mean? You get infected, virus remains in hiding without replicating in some parts of your body. That's called latent infection. Now, it can. I'll tell you a little bit more about that later. And we've looked at fatal disease. Can viruses be transmitted from mother to child?
Yes,
Yes,
sir.
sir.
I think so.
So HIV is
Yes.
transmissible if I'm not wrong.
Yes, it can be transmitted. So that's called congenital disease. Can viruses cause anything else important? Very important. We all know about this.
So can cause cancers. I guess
That's
EBV
correct.
causes Hodgkin's
Viruses
lymphoma.
today, as of today, viruses cause about 1/4 to 1/3 of all known cancers are related to viruses. So that's all I have in this slide. I think all of you would be able to see it. So we looked at the example of common flu for this. We looked at the example of HIV for persistent disease.
So we are not able to see.
Okay, you're not able to see anything. Okay. Okay, give me a second.
It is just consequences of viral infections, and after that we are not able to see anything.
So I could see. So probably it's
I
rough
could
nature
see the
internet
screen.
connection.
So the rest of us could see
I
it.
can also see.
Okay, I tried to redo it. Can you all see it? And can you see my pen moving? Or can you see it?
Yes,
Yes,
sir. I
sir.
can.
Okay. For latent
Yes, sir. Now I
infection.
can see it.
Okay, good. For latent infection and reactivation, we looked at the example of chicken pox. Once you had it, the virus, the virus remains hiding in the nervous system, and at the opportune moment when your immune system is suppressed, it can get reactivated. So that's called latency and reactivation. Viruses can lead to fatal disease. For example, sometimes in SARS-CoV-2 also it can happen. Ebola and other diseases, other viruses can lead to fatal disease, much higher rates of fatality. Congenital disease from mother to child, good example is HIV. Many other examples, we look at it. Several cancers are caused by viruses. Can anybody give me an example of a virus related cancer?
So Hodgkin's
HPV.
lymphoma.
HPV is good. Any other example? One more example, if anybody can tell me. Hepatitis B virus causes liver cancer. Anybody else?
So
Epstein-Barr virus.
Epstein-Barr virus, Hodgkin's lymphoma.
Very good. Epstein-Barr virus can cause a variety of cancer, including nasopharyngeal carcinoma and some types of lymphomas. Okay, so that's Epstein-Barr virus. So viruses can also contribute to other diseases. Can anybody give me an example of another disease caused by virus? Anybody? Other than cancer?
So other diseases as in like other groups of diseases or like
No,
name
no.
disease such as dengue?
No, not like that. Okay, maybe I think the question is not very clear. They contribute to other diseases. For example, HIV can lead to immunosuppression, which can lead to a variety of other diseases. There are some
Yes, sir.
viruses which are associated with type 1 diabetes in young children, type 1 diabetes mellitus in young children. So viruses can also, some viruses can cause liver disease, some viruses can cause uh, neurodegeneration, uh, some viruses can affect the heart. They can contribute to a variety of other diseases, just depending on where they can replicate or what kind of immune response they elicit from the body. They can contribute to other diseases as well. Somebody was asking something, sorry. Did any
No,
of
so
you want?
I was
Go
just
ahead.
extending your HIV example that you have given. It will lead to fungal infection due to immunosuppression of the body.
That's correct. So basically, what I was expecting is, although HIV can lead to a lot of things, one of the major things it contributes to is general severe immunosuppression. Hepatitis viruses, many of them can lead to liver diseases and liver damage, for example, in the long run, and liver cirrhosis, for example. They can lead to other liver diseases also. They can also lead to other autoimmune diseases. They can be a contributory factor to other diseases. That's all I meant. Is that clear?
Yes, sir.
Okay.
Yes, sir.
Okay, so here I've summed up whatever we just discussed. In all of you know what we discussed here. So virus infections can be acute, chronic, or latent. When you say acute, it means that in 6 months, usually the body will be able to fight and eliminate it. Most of the infections that we talk about fall into this category. Of course, there will be virus in the body. It will be replicating. It can cause disease. Sometimes you can have asymptomatic infection. That's why you have a minus here. You may or may not have disease. So chronic and latent infection, both times the virus will be present in the cell, but they are there for several years, as you can understand. That's why they call chronic or latent. What is the difference between chronic and latent? When I said latent, the chickenpox example, the virus is not replicating in the body. It's not replicating in the body. It's just hiding in some cells. Okay, and it's not even causing disease. Like the two of us who have had chickenpox, the virus is hiding in our body. It's not causing disease. So it's not replicating either. So that's called latency. When we say HIV, for example, the person's body makes millions of HIV every single day. So the virus actively replicates. This is for several years together, 10 years, 15 years together. There is active virus replication. There may or may not be active disease, because sometimes HIV infected patients, they have 4, 5 to 10 good years of life. After that, their life deteriorates because they have more profound immunosuppression. So it could be asymptomatic as well. So the difference between chronic and latent is that in chronic infections, viruses replicate. In latent infections, viruses don't replicate. They're present but remain in hiding. So that's the difference. I'm going to come back to these two slides in a second. So sometimes I just have it this way, because if there isn't enough time, I stop there. But today we do have enough time. I'll take only 10 more minutes and we'll try to finish the rest of it. So what about virus pathogenesis? All viruses cannot infect all organisms, and you know it. And you know this, for example, SARS-CoV-2, it is believed to have come from a bat virus or some related pangolin virus. So they do have a narrow host range. Apart from SARS-CoV-2, can anybody give me an example of a virus that can infect at least 2 different animals?
Ebola is an example which came from primates.
Okay. Any other much more
Influenza
common?
virus.
Huh?
Influenza virus, sir.
Influenza virus. Anything else that's much more common in IITD than outside IITD?
Rabies.
So dengue.
Rabies, okay, rabies is not common. The dogs are more common on campus, that's correct. Dengue
So
virus
dengue.
is a good example because it can infect
Report virus.
mosquitoes and humans, right? So that's an example. So when we say host range, it means the animal or the organism which it can infect, and within the organism or the animal, usually viruses infect one or only a few tissues. They can't infect all the tissues. Now, what determines which tissue is infected by the virus? The presence of a specific receptor on the host cell, on a particular type of host cell, determines whether the virus can get in or not get in. So now all of you read a lot of news. What is the receptor for SARS-CoV-2?
ACE2 and TMPRS22.
That's right. So there are some molecules that proteins, host cell proteins on the host cell surface.
ACE2.
ACE2, yeah. So these proteins have other functions, but fortuitously or accidentally, the virus makes use of that to get into the host cell, and that makes human beings susceptible for SARS-CoV-2. If we did not have that ACE2 expression, we probably would be resistant to SARS-CoV-2. So just an example so that you can relate to what host range means. Now, overview, I'll take only a few more minutes and we'll stop. How do viruses replicate? They attach to a receptor on cell surface. They get into the cell, that's called penetration. And once they get into the cell, everything is dismantled. This is called virus uncoating or disassembly. And after that, all the parts are synthesized. More nucleic acids are synthesized, more virus proteins are synthesized, and the virus gets assembled and then released. So then hundreds of thousands of new viruses are released from an infected cell. These are the stages in which a virus replicates inside a host cell. Now, one or two general features about virus replication. So when, for example, I'll perhaps give the example of poliovirus, which is the best example to understand virus pathogenesis. Now, what does poliovirus cause? Anybody? Guys? Nobody? I know we have eradicated polio, but only recently. Go ahead.
It is acute flaccid paralysis.
Very good. It's very precise term. It causes something called acute flaccid paralysis, but without going into details of that, basically it just causes paralysis, usually of the lower limb. At least one lower limb is usually what is affected. Okay, so that's poliovirus. So and it is very common, almost like like it used to be such a threat. Now we've eradicated it from most parts of the world except some parts of Asia, including Pakistan, and some parts of Africa. Everywhere else in the world, polio has been eradicated. And this happened not too long ago. This happened when you guys were in high school or around that time only. It is not too long ago. Actually, it happened only about 5 years, 6 years ago. So that's when this happened. We eradicated polio. Now, how does one get polio? The paralysis is because it affects some parts of the brain and kills some neurons. That's why paralysis happens. But let's see how the virus gets into the body and how it replicates. I'm not trying to teach you poliovirus and its details in today's lecture. I'm using poliovirus as an example to understand overall aspects of virus pathogenesis. Excuse me.
Sir,
Yeah, go ahead.
sir, I had a question like how did we eradicate polio from all over the world?
How did we eradicate it?
Yes, sir.
Okay, this is because we used a highly efficacious vaccine. There are two types of vaccine, live attenuated and killed vaccines. The one that we use in India, the oral polio drops, is a live attenuated vaccine. And we had very effective campaigns, like the entire North India would be vaccinated at the same time. Entire South India would be vaccinated at the same time because these oral polio drops do not require expertise to administer. Any Tom, Dick and Harry without any education can also be a social worker who administers this. So when they had coverage, door-to-door coverage of vaccination, pretty much the entire country got vaccinated. And once we get vaccinated, we get protection. And there's something called herd immunity. We'll talk about it later. And then basically we eradicated all the cases, because if nobody is shedding poliovirus for some time, there will be no more new cases of polio in India. It can come into India from if other people bring it. Now those other people are limited to few countries in Africa and one or two countries in Asia. Other than that, all the countries have eradicated it by using this vaccine. So poliovirus vaccination is a very interesting topic, and it is the best topic to understand vaccinology in virology. Best example to understand is poliovirus vaccinology. We'll talk about it in great detail later. But short answer, highly efficacious vaccine with good public health programs to administer it at large scale helped us eradicate it. Okay, now going on to understanding of general virus pathogenesis in 2 or 3 lines only. Poliovirus affects neurons to cause paralysis. But how does it enter the body? It enters the body through the respiratory tract. It replicates first in the tonsils in your throat and then a little bit in your stomach. From there, it enters the bloodstream. From there, it enters your bloodstream, and from the blood, it spills over into the central nervous system. Give me a second. It spills over into the central nervous system, where it can affect the brain and cause damage to some neurons, leading to paralysis. Now, the replication of poliovirus in the throat and the stomach, which happens in any infected individual, is called the primary site of replication. By systemic spread, it means that when it spills over into the blood and is taken to other organs by the bloodstream, including the central nervous system or CNS, that's called systemic spread, which means spreading throughout the body through a medium. Usually this is most often it happens through blood. Sometimes the systemic spread can happen through neurons. So these are the ways by which the virus can spread outside a local environment when it infects somebody. Now, what is the secondary site of replication here? The secondary site of replication here is the neuronal tissue, which causes the actual disease. So the primary site is the throat and the stomach. Spread is through blood and CNS. And the secondary site of replication is neurons, certain kind of neurons. So that's the secondary site of replication. You don't have to, this example is just so that you understand it. Many viruses have a primary site of replication. They can enter the bloodstream to spread throughout your body, and they may have secondary sites of replication. In this case, it's the CNS. So this is generally how viruses cause diseases.
Sir?
Give me 1 second.
Sir.
Just one
Yes,
second.
sir.
I'm getting a call. Hi, James. In 10 minutes. Okay. Okay, sorry about that. Go ahead and ask your question. Sorry.
Sir, my question was, what helps them to spread through the central nervous system?
Okay.
What is the mechanism? Like blood stream, through blood stream they can spread. That is possible, like that we can understand. But what helps them to spread through central nervous system?
Okay, so once anything is in your blood, especially something small like a virus, especially something very small like a polio virus, can easily cross what is referred to as the blood-brain barrier, or BBB, which is a very selective network. It only allows some things to cross over from the blood to the brain. For example, you need glucose. It doesn't allow the entire blood to go there. It only allows exchange of many things, exchange of several things. Not everything in your blood can cross the blood-brain barrier. Okay, so viruses can cross it, especially small viruses. Once they cross it, if certain kind of neurons have the receptor that is necessary for poliovirus replication, then they can infect those, and then they can do a cell-to-cell spread within the central nervous system. Does that answer your question?
Yes, sir. Yes.
Okay. So we'll look at all of these in great detail. Don't worry about it. Last one or two slides. How do you cultivate viruses? Anybody? How do you cultivate
If we have
viruses?
to cultivate, we will have to use like plant cell, animal cell, or bacterial cell, in whichever they can grow.
Okay. Partially right. Okay, we can use something called cell lines, which are usually human or animal cells. If you're looking at plant viruses, you can use plant cells. So these cells are grown in the laboratory. Usually these are cancerous cells, and they can be grown in the laboratory in an uncontrolled manner. They can keep on replicating. There is no limit to their ability to proliferate. So these are called cell lines. You can put them on Petri plates, you can put them on flasks, they can grow well. And because they are derived from the tissue of interest, for example, some liver cells support the replication of hepatitis viruses. Many cells in the body support the replication of polioviruses, herpes viruses, chickenpox virus. So we use appropriate cell lines to grow them. You can also use embryonated eggs, which means eggs that contain an embryo because they have yolk and other nutrients. They support virus replication very well. Not all viruses can grow in all these systems. For a given virus, you have to use the specific system. But I'm just trying to tell you here the overview of how you can grow viruses. The other way is to use animal models, rabbits, mice, monkeys, etc., to grow viruses in the laboratory. So these are the ways by which you can grow viruses in the laboratory. I think last one or two slides, and then we'll go to the criteria and close. So give me a second. Hello. நல்லா இருக்கும் கிளாஸ்ல இருக்கேன் கூப்பிடுறேன் திருப்பி. Sorry. Okay. So in terms of antibacterial agents, how many of you have taken an antibiotic in your life? Almost all of you, right? Without exception.
Yes, sir.
That's because there are many antibiotics, and they are highly selective because bacteria has its own machinery to make proteins, to synthesize nucleic acids, and they can be targeted, and they are different from the human system of making nucleic acids and proteins. So that's why we have a lot of antibiotics. But however, viruses use host cell metabolism and host cell apparatus to make the virus proteins. Virus nucleic acids are also, they use our nucleic acids, they just copy their genome. So that makes it very difficult to target viruses because selectivity becomes an issue, and because selectivity is an issue, you might start interfering with the host processes, which is our own cellular processes, if you try to target viruses. So antivirals are difficult to design compared to antibacterials in some way. Now, why do we even need antivirals today? Because there are many diseases which are emerging diseases. You know a good example of that. Vaccine development may take several years. Before that, antivirals may help. Some viruses, such as HIV, have been around now for 4 decades or more, but we don't have a vaccine for it because they are very rapidly mutating. So you need antivirals to control them. So, and there could be problems with vaccine coverage like we are having right now. They're saying for Omicron, the efficacy of the current vaccines is poor. So you could have issues like that. And you could have individuals such as immunocompromised individuals who may not be able to take vaccines because live attenuated vaccines cannot be given to them. So they will need antivirals And, and for a variety of reasons, we do need antivirus. I think this is the last slide I have before I go on to the criteria for evaluation. So what about what about antivirus? Which stage of virus replication do they target? I have it here. They target this stage. Most antivirus that we know of, almost 95% of all antivirus we have today, they target the synthesis of virus nucleic acids or synthesis of virus protein. That is the step that they target. And most antivirus that we have today are against 4 or 5 viruses only. Most other virus infections, we don't need antivirus because they are acute self-limiting. We don't need. Only for viruses which are associated with long-term or chronic or persistent infection, we need antivirals. Good example is HIV, hepatitis B, hepatitis C, and some other deadly viruses. For example, influenza virus. If you're a high-risk person, you might need antiviral agents. There are antivirals for influenza also. These are some examples of viruses against which we actively use antivirals. There are only a few more viruses against which we use antivirals. Most viruses, we don't use antivirals. Any questions until this point of time?
So why not target the first like step or the adsorption step?
Okay, why not target that? So if you target that, the way it'll work is to block the receptor, right?
Yes,
Does
sir.
that make sense? Let's say ACE2 is the
Yes, sir.
receptor. You block ACE2, but ACE2 has other important functions. And ACE2 is
Right.
present in many other cells in the body. You
Right,
understand
sir.
what I'm saying?
Right, sir.
You might be blocking an essential function that is necessary for at least some cells, if not all the human cells, and that might lead to a variety of side effects. Does that make sense?
Yes, it does.
So typically now there are approaches that are coming up like that, but side effects are a wait and watch. We don't know. So I'll quickly wrap up in the next 2 minutes or so. Uh, why you want to study human virology? What will you be studying in this course? In this course, you'll be studying about excuse me, virus structure, function, replication, how it causes disease, how do you diagnose it, how do you treat it, how do you prevent these diseases. Basically, it gives you an overview of important human viruses and details of how they cause disease, how to diagnose them, how to prevent them, etc., along with some fundamentals of virus structure, function, replication, etc., some basics, antivirals, how do vaccines work, what are the different types of vaccines, and with each virus you'll understand what test to diagnose, what sample to collect, and why it's important to do that, why diagnosis is important, why prevention is important, how prevention can be done. These are the things that I'll be covering in this course. We'll be looking at important human viruses only. We won't be looking at plant viruses or other animal viruses. And this is the contents of the course, basically the same stuff. At the end of the course, what I want you to appreciate is if you read a paper related to viruses or anything to do with viruses, let's say you're working on protein biochemistry, you can still apply this if you understand the fundamentals of virus infection and virus pathogenesis. Basically, that is the idea, to give you so that when you read something, you should be able to understand the basics of what you're reading related to viruses. If you're PhD students, I'll be really happy if you try and apply this to anything that you're doing. So that is the aim of the course. What is the evaluation policy? We'll have a minor and a major, 35 marks for the minor, 40 marks for the major, and there'll be a term paper or some sort of an assignment or a presentation that will carry 20 marks and up to 5 marks for attendance. if you if you don't have 50% attendance, typically we do 1 grade less. If you have, unless you have a medical reason or something, we'll do 1 grade less. If you want to audit this course, you need only 35 marks and 50% attendance. I think that's all I have. If you have any questions, we have another 5 minutes. Hello?
Sir, can we learn in this course like transformations and that regarding the viruses?
Say that again. Transformation of what?
A transformation, like the mechanism of transformation, according to which genetic materials can be transferred into like bacteria or other cells.
Uh-huh.
Yeah.
There would be some examples of how to use viral vectors and their applications.
Yes,
That
sir.
would be
I am
only a
just talking
small
about
part
that.
of a lecture. Virology is much broader than that. But
Yes, sir. I just wanted to know that will that topic be covered?
Yeah. There are some aspects of viral vectors which will be covered in this course. Does that answer your question?
Yes, sir. Yes.
Okay. Anybody else has any questions in terms of evaluation policy or anything else about today's class?
So just one thing. So could the attendance marks be counted post like the ad drop period? Because I guess a friend of mine wants to take
I
up
always
this
do
course.
that. I always do that. Don't worry.
Thanks a lot, sir.
Sir, in case of acute infection, it was mentioned on the slide that diseases is plus or minus.
That's correct. Okay.
So
How
when is
can
this
that
minus?
happen? Okay. A good example is Omicron cases that you're looking at. Many of them are asymptomatic. Somebody just told me a group of 80 college students went for a tour. Pretty much none of them have symptoms, but all of them tested positive for Omicron. You understand what is asymptomatic?
Yes, sir. Got it.
Yeah. I understand where your question is coming from. It doesn't mean that the virus is not replicating. The virus is replicating, but it's causing such minimal disease or damage to the tissue, and it's not even spreading very well systemically, that you don't have any tangible symptoms. And that's why it's asymptomatic or mildly symptomatic. Does that answer your question? Sometimes, actually, when we have a common cold, it could be very mild or close to asymptomatic. There isn't anything, but you might sneeze a couple of times and you're good. You see what I'm saying, Siddharth?
Yes, sir. I got that. Thank you, sir.
So it also includes mild non-specific symptoms, because sometimes when you have mild fatigue, you don't know whether it's virus or not. It could be just exertions, things like that. It's a little bit of a gray area. You don't have strong symptoms, you don't have specific symptoms. Many virus infections can lead to something like that. For example, I can be infected with some diarrhea causing viruses. I might just not feel hungry for a meal, but other than that, everything else may be fine. I may not have the real diarrhea, but somebody else who gets the same virus may have diarrhea. So then I become asymptomatic, the other person becomes symptomatic. You understand what I'm saying?
Yes, sir.
Okay. Anybody else? Any other questions? Okay, guys, I'll send you the slides at the end of the class. Sorry, at the end of every week, I'll send you the slides. And if you have any other questions, you can contact me. I'll also tell you the pattern of the question paper and other things subsequently. Some of you have been in my course, you know the pattern. For the others, I'll just detail that also. Before the exam, I'll detail the patterns of the question paper as well. Okay. Thank you, guys.
Thank you, sir.
Thank you, sir.
Thank you, sir.